Hello, in this class, we will take a look at bacteriophages. Bacteriophages, or simply phages, are the viruses which infect bacteria. What you're seeing here is a typical bacteriophage. These are viruses which infect bacteriophages. You can see here a SEM picture. This is a cell of a bacterium, and you can see multiple number of viruses that are infecting this bacterial cell. Let's get along with bacteriophages. Bacteriophages are of different types depending on the genetic material and the type of the genetic material that is present with them. They are mainly of 4 types. They are single-stranded DNA phages, double-stranded DNA phages, single-stranded RNA phages, or double-stranded RNA phages. Single-stranded DNA phages include the 5X174 phage, the 5R, the G viruses. Double-stranded DNA phages includes the T-even phages and the T-odd phages. Single-stranded RNA phages include the MS2 and M12, and double-stranded DNA phages example is 5G. So this is based on the type of nucleic acids and the type structure of the nucleic acid present within the bacteriophages. Bacteriophages were first discovered by Joshua and Esther Lederberg, and they are mainly of 2 types, the lambda phages and the T phages. And these are bacteriophages which infect E. coli or Escherichia coli. And the phages have a very complicated structure, or one of the most complicated structures that you can see in viruses belongs to bacteriophages. Mainly the bacteriophages, the structure comprises of a head and a tail. And in this case, the head is an icosahedron, that is, it is polyhedral in shape, made up of icosahedron of 20 faces and proteins of several types. And it contains the genetic material, which is a DNA in this case, which is around 46,500 base pair long. Then it has got a protein tail, which consists of around 35 stacked discs, which end in a tail fiber. So this is the structure of lambda phage. So lambda phage is having a head protein within which you can find the genetic material, which is a DNA. Then it has got a protein tail comprising of 35 stacked discs, and it ends in a tail fiber. Lambda phages are important since they are being used to construct genomic DNA as well as cDNA libraries in genetic engineering. Moving on to the most complex type of phages, the structure, most complex structure in viruses belongs to T-even phages. So here again you can see, just like in the case of lambda phages, T-even phages also have a head and a tail and the genetic material. Here, genetic material is again DNA, but look at the head. It is again an icosahedron with 20 different phases, but the tail is a little bit complicated. The protein tail has got a tail sheath which ends in a basal plate, which have small little spikes projecting out and long tail fibers. This basal plate, the spike, and the tail fibers help to firmly attach the bacteriophage on top of E. coli cells, and the icosahedron head protects the genetic material with them. So this is the structure of the bacteriophages. So you can have short essays which will ask you to describe the structure of bacteriophages with neat labeled diagram. So once again, a little recap on the phage structure. The phages, bacteriophages, are viruses which infect E. coli or the bacterium, and they have got a head and a tail. In case of lambda phage, the head is an icosahedron with 20 phases, and the protein sheath comprises of discs of protein stacked one on top of other, almost around 35 numbers, and it ends in a tail fiber. It's comparatively simple. But the T-even phages, it's a little bit more complicated. Again, the head is made up of icosahedron with 20-sided capsids, and then the tail sheath ends in a basal plate having little spikes and long tail fibers. So this is the structure of bacteriophages. Now let's see how they replicate. Just a recap of the infection stages and how they multiply. We have seen this in earlier class. The bacteriophage comes, lands, this single line that you can see here, it represents the surface of the bacterial cell. It comes, lands in the bacterial cell, then attaches itself with the help of the basal plate, the spikes, and the tail fibers in case of T phages, and it pins itself or absorbs itself onto the surface of the bacterial cell. Then slowly it starts penetrating the surface membranes of the bacteria and injects the viral DNA into the bacterial cell. Let's see what happens once the viral DNA enters into the bacterial cell. Bacteriophages can be classified into two based on how they multiply within a bacterial cell. Based on the mode of multiplication, bacteriophages can again be divided into two, the lytic phages or the lysogenic phages. Lytic phages are also known as the virulent phages. Lysogenic phages are also known as non-virulent or the temperate phages. Let's check out each. Lytic phages or the virulent phages multiply through a cycle known as the lytic cycle, and the lysogenic or the non-virulent or the temperate phage multiplies itself through a phase known as the lysogenic cycle. Let's check out each in detail. First, let us see what happens during a lytic cycle. As we have seen earlier, the bacteriophage, as you can see here, will come, attach itself onto the surface of the bacterial cell, absorbs, and then finally penetrate and inject its DNA, viral DNA, into the host cell. Once this viral DNA enters the host cell, this viral DNA takes over the machinery of the bacterial cell, and it disintegrates or chops down the bacterial chromosome and takes over the whole cell. Using the system or the biosynthetic machinery present within the bacterial cell, the genetic material of the virus divides, multiplies. It also synthesizes the cod proteins in the stage of biosynthesis. Once biosynthesis is complete, assembly takes place where the multiplied genetic material of the virus gets packed up within the viral cod proteins, and the daughter virions are now ready. Once the daughter virions are ready, it bursts open the cell wall of the bacterial host and gets itself released outside. So what happens during lytic cycle? The bacterium comes, absorbs onto the cell surface of the bacteria, injects its viral DNA into the bacterial cell. The viral DNA takes over the whole machinery of the bacterial cell, uses the biosynthetic machinery of the bacterial cell to replicate its genetic material as well as produce the cod proteins. Once the genetic material is replicated and the cod proteins are produced, assembly takes place, mature virions are formed, it breaks open the cell of the bacteria, kills the bacterial host, and is released outside. So this is what happens in lytic cycle. So the T phages follows lytic cycle, and they are known as the virulent phages. In case of the temperate phages or the non-virulent ones, it follows lysogenic cycle. So what happens in lysogenic cycle? In this case also, this is being followed by lambda phages. The phages comes, lands in the bacterial cell, injects its viral DNA into the bacterial cell. But unlike in lytic cycle, the viral genetic material does not completely disrupt the bacterial genetic material, but it integrates itself along with the bacterial DNA to form what we call as a provirus or a prophage. This provirus or the prophage will not directly kill the bacterial cell, but instead it gets divided itself whenever the bacterial cell divides. So as long as it is seen integrated with the bacterial DNA, this prophage does not harm the host. It keeps on dividing itself as and when the bacterial cell divides. But there are certain times when it gets certain shocks, maybe some nutrition deficiency or a temperature difference. At that point of time, what happens is this provirus or the prophage undergoes a process known as induction. And during induction, what happens is the viral genetic material gets itself detached from the bacterial DNA and then it enters into the lytic cycle. So once the lambda phage attaches to the bacterial cell, it injects a genetic material into the host cell. This genetic material, viral genetic material, gets attached itself to the bacterial genetic material to form what we call as the provirus or the prophage. The provirus or the prophage gets itself multiplied many number of times as and when the bacterial cell divides. But once it gets some shock, the provirus detaches itself into the viral DNA and the bacterial DNA. Once this process or the induction process takes place, this now enters into the lytic cycle. In the lytic cycle, biosynthesis takes place, assembly takes place, and finally the virions are released by disrupting the bacterial cell wall. So this is what happens in lysogenic cycle. So what are the major differences between lytic cycle and lysogenic cycle? First thing is, lytic cycle, the genetic material does not get integrated with the bacterial chromosome, but instead it will disrupt the bacterial chromosome, takes over the cell machinery, then it bursts open, kills the bacterial cell and comes out. In lysogenic cycle, what happens? The viral genetic material gets integrated with the bacterial genetic material to form what is known as a provirus. Here it is not disrupting the bacterial cell as such, but once induction takes place, it enters into the lytic cycle and finally kills the host and comes out. So this is what happens in lytic cycle and lysogenic cycle. There are certain terminologies associated with the lytic and lysogenic cycles. You have to remember certain temperate phages. What are they? They are phages which follow the lysogenic cycle. Virulent phages are those which follows the lytic cycle. Then you need to remember prophage. What is a prophage? It is a viral genome which is integrated with the bacterial genome. You need to remember the word induction. That is a process by which a prophage separates into the viral genome and the bacterial genome. Then you have lysogens or the lysogenic strain. These are the host bacteria within which lysogenic cycle takes place. Then lysogenization is a process, transformation of a normal bacterium to the lysogen by the viral infection. Then lysogeny is a close association between the viral genome and the bacterial genome during lysogenic cycle. And the lysogenic state is a symbiotic association developed between the phage and the bacterium during lysogenic cycle. And this symbiotic association results in the formation of prophage. So to summarize, we learned about bacteriophages, which are the viruses that infect bacteria. We spoke about the structure of bacteriophages. They are complex structures with a head and a tail and the genetic material. We discussed about the structure of the lambda phage as well as the T phages. Then we spoke about the multiplication in bacteriophages. They can be either through lytic cycle or through lysogenic cycle. The phage which follows the lytic cycle are known as the lytic phage or the virulent phage. Example is a T phage. Lysogenic cycle is being followed by the non-virulent or the temperate phages. For example, that takes place in lambda phages. The major difference between the lytic and the lysogenic cycle is that In lytic cycle, it leads to the immediate death of the host, while in lysogenic cycle, immediate death of host does not take place. But on the other hand, in lysogenic cycle, there is an integration of the phage DNA into the bacterial DNA, which results in the formation of a raw phage. So we will move on to the other types of viruses and the other infectious particles in the next class. Thank you.